Welcome student. This is Vikas Sampatorpe, working as Assistant Professor, Department of Zoology in GMD Arts, BW Commerce and Science College Shillong. In this video lecture, we are going to start antibodies, their structures and functions. Normally inside the human body, the antibody present in the form of serum, and this serum is a serum glycoproteins. Okay, inside of human body, near about 80% is the serum glycoproteins, and this serum glycoproteins is IgG antibody. IgG antibodies is present in the inside the human. Okay, they are also play the major role in immune response. That means they are the synthesis of immunoglobulin inside the body. Here the another function, adaptive immune response. Okay, that means they are play the major role in antibody synthesis. We are also called as the humoral immune response also. Then the next part is antibody structures. These antibody structures are made up of two light chains and two heavy chains. Okay, the light chain is a 25 kilodalton and the heavy chain is 50 kilodalton. Okay, but both chains are identical here. Okay, the difference between here only there are the 25 kilodaltons. Okay, and the heavy chain is 50 kilodaltons. Then the next point is light chain bound to the heavy chain by the disulfide bonds, and the heavy chain is bound to the heavy chain with the disulfide bond. Okay, these are the two important sentences here. The first sentence is a light chain bound to the heavy chains with the disulfide bond, and heavy chain bound to the heavy chain with the disulfide bonds. The difference only here, in a first sentence the heavy chain bound to the light chains, and the second sentence heavy chain bound to the heavy chains. Okay, then the next one is the first 100 amino acids. This first 100 amino acid terminal is vary for both heavy and light chains and are variable, and that's why this region is also called as a variable regions. Then next one, this one is also called as a variable light regions, variable heavy regions, constant heavy regions and constant light regions here. Then here the one regions is present, and this region is called as the CDR region. What is meant by the CDR? CDR means the complementary determining regions, and this complementary determining region which is useful to the binding of antigens, and normally in the structures of antibody here the two CDR regions is present. That means the two antigens is attached to the antibody structures. Okay, then the last point here, the remaining regions are very similar within the same class. Okay, this one is a repeating domain. Here the one repeating domain is present, and this repeating domain near about 110 amino acids. What is the function of this repeating domain? This repeating domain is interchange disulfide bond with each domains. Okay. Here the second point, the heavy chains. One variable heavy chains are present here, and either 3 or 4 constant heavy chains. Okay, the one is a variable heavy chain, and either 3 or 4 the constant heavy chain is present here. We are also called as constant heavy regions 1, constant heavy regions 2, constant heavy region 3, constant heavy regions 4 also. Okay, then the third point here, the light chains. Here the two light chains is present. One is a variable light chains, and second one is a constant light chains. Okay, the third point here, a hinge region. This is a very important part of the structures of antibody, and this hinge regions is a flexible regions. This hinge regions is a flexible regions, and these flexible regions are the proline rich residues are present, and that's why they have the flexibility there. Okay, then the second point in hinge region, the hinge is found in only IgG antibody, IgA antibody, IgD antibody. Okay, then The proline residue are targeting from proteolytic digestions. Okay, like as the papain and pepsin here. Okay, then the next point here, a rich cytosine residue are present here, and this cytosine residues, that's mean the simply a disulfide bond here. Okay, then another antibodies here, one is the IgM and second one is the IgE. This one is absent of hinge regions. Okay, that's mean the hinge region only found in IgG, IgA and IgD antibody, and this hinge region is totally absent in the IgM, IgE antibody. Okay, IgE hinge regions. Okay, and the last point is they are instant have the extra CH4 domains are present here. Then the next part is this one is the actual structures of antibody. Okay, this side is a just a minute. This side is NH3 regions is present and this side is the COO minus region is present. Okay. This one is the N terminal and this one is the C terminal. The N terminal amino group is present and the C terminal carboxy group is present. The C regions is the carboxy group is present and this N terminal is the amino group is present. Okay. This amino group is attached to the variable light chains, okay, and this carboxylic group is attached to the constant light chains. Okay, this one is a totally light chains. Okay, on this sides here one another amino group is present, and this sides here one another carboxyl group is present. That's means this is the N terminal of light chain and this one is the C terminal of light chain. Same as it is on this side, this one is the N terminal of heavy chains and this one is the C terminal of heavy chains. Okay, then then here this one is a constant light chains and this one is a constant heavy chains. This constant light chains and this constant heavy chains are attached with each other with the help of a disulfide bonds. Okay, this variable regions and variable light regions and variable heavy region does not attach to here with the disulfide bonds. Okay, the total numbers of amino acid in this region, particular region, here 240 amino acid residue in a light chain and in a heavy chains here 446 amino acids are here. Okay, this variable light chains also made by the 25 kilodalton and this constant light chain also made up by the 25 kilodaltons. Okay, the molecular weight 25 kilodaltons here and 25 kilodaltons here. Okay, this is the heavy regions and these heavy regions are here the CH1 group is present. Okay, CH1 group is present and here the CH2 group is present, here the CH3 group is present. These are the three regions is present. We are also called as constant heavy 1 region, constant heavy 2 regions and constant heavy 3 regions are here. Okay, these are the three regions and these three regions are made by the CHO group, that's mean the carbohydrate, CHO carbohydrate region. Okay, this one is also carbohydrate region. Okay, here the important part is the hinge regions. What is the hinge regions? Hinge regions is a proline rich regions and this hinge regions is a flexible region. Here the ability to flexibility. Okay, here the these regions are able to flexibles. Okay, then on this side the same concept here. Okay, here also variable heavy regions, variable CH1, CH2 and CH3. Okay, again here the carboxylic group, C terminal of heavy chain and this one is the N terminal. Okay, okay, same as it is here, this one is the N terminal and this one is the C terminal. Okay, this is the first heavy chain on this side, second heavy chain. Okay, this one is the first light chain and this one is the second light chains. Okay, And we know that the structures of antibody are made up by two chains. One is a heavy chains and second one is a light chain. And two heavy chains are present here and two light chains are present here. Okay. This site is also known as antigen binding site. This one. Okay. This variable heavy regions and variable light region. In between these regions here, the one gap is present and this gap is filled by this gap which is filled by the antigen and antigen attaching site. We are also called as a fab region. We are also called as a fab region, and this fab region, okay, F A B, that means the fab region, and this fab region, the antigens are coming and bind here. Okay, the same size here, one more fab region is present, and same the functions of antigen binding here, and this one is the actual antigen binding site of these structures. Okay, and that's why this region is called as a fab region, antigen binding site. And this region shows the biological some biological activity. In this region is called as a FC region. This region is also called as the FC region. This is the detailed structures about the antibody. Then this one is the second structure, and this is the actual structures of antibody. Okay. Here total numbers of amino acids. Total numbers of amino acids. This one is our N terminal. This one is our C terminals. Okay, N terminal and C terminal, same as it is for this sides. This one is the N terminal and this one is our C terminals. Okay, total numbers of amino acids here 214 amino acids. Okay, in between the one fold, in between the one fold, the folding is start for the 22nd amino acid and end for the 88 numbers of amino acid. Okay, this one is the first foldings on the heavy chain regions, and this side is the second folding on the heavy chain regions. Okay, here the folding is start from the 144 amino acid and end for the 100 and 200 200 amino acids. Okay, this one is a very important concepts. Then here another one fold coming from the 261 amino acid, and this one is completing the 321 amino acid. Next folding is 367 amino acid, and complete this folding 425 amino acid, and total numbers of amino acid is 446 amino acids. Okay, same for this sides. Okay, this side is the antigen binding site and this side is the biological activity side. Okay, and this whole about the detailed structures of antibody. If you have any questions, please ask on directly. Thank you so much everyone.